So next we have Daryl Chastain, who is an SY on the USDA side. He's gonna introduce himself as part of his talk. I guess I need to share again. All right, take it away.
Okay, so there's the arrows, right?
Yeah.
Okay, hi, I'm Daryl Chastain. Good morning, everyone. A little bit about myself. I was born in Fort Smith, Arkansas, married in 2006. I have 2 children. I live in Greensville. I have a Bachelor of Science in biology from the University of Central Arkansas, Master in Biology from the same university. Mostly I studied heat tolerance and drought response in cotton. For my PhD, I went to Georgia and worked in drought response in cotton and a lot of leaf characteristics and irrigation scheduling. So for my talk today, it will be drought response modeling of leaf photosynthetic parameters in 2 Gossypium species. So a little bit of background for upland cotton. Whenever you experience a drought, pretty much within all the commercial cultivars, you're going to have the same response. You're going to have a overall decrease in net photosynthesis. Midday respiration stays about the same, but being that net photosynthesis decreases, the ratio of the 2 actually ends up increasing. Parameters related to water movement and evaporation, like stomatoconductance and transpiration, tend to decrease as water stress increases. Because of this, the cooling that the leaf normally goes through is inhibited, so you end up raising leaf temperature, and we use that a lot for irrigation scheduling. Some of the primary photochemistry, like photosystem 2, which I'll discuss in just a moment, seems to not really be affected, but there are other photosynthetic parameters that are. Photorespiration, or basically wasted carbon fixation, it ends up, that's where your decrease in net photosynthesis tends to come from. But a lot of the energy capture processes, there's a hot debate about what's going on, what's really happening. 20, 30 years ago, we thought we had it figured out, and then, people like me come through and break it apart. So the primary photochemistry is the light capturing processes in cotton, or in a plant in general. So there's a laser on this thing. Let's try this. This portion right here is where most of the work has been done over the past 20 or 30 years, because it's really easy to measure. This process seems to be virtually unaffected by drought. And that caused a lot of arguments because there are other ways to estimate the stuff. So chlorophyll fluorescence, briefly, the way it works, if a light molecule, or a photon rather, hits photosystem 2, there's 3 fates it can go through. It can either be dissipated as heat through non-photochemical quenching or just direct transmittance of heat. It can be reflected and go right back in, like certain wavelengths like green, that's why plants are green, or it can go into photochemistry. Well, if you can bombard photosystem 2 with a high energy light, maybe 4 times what a solar maximum in a day would be, you kind of overwhelm the system and you can get that fluorescent signal boosted, so you can get a lot of information on how energy is transferring through that system. Newer techniques, you can follow each energy transfer step and get a quantum efficiency as it moves, as an electron moves through the thylakoid membrane up to the point where it ultimately produces ATP and NADPH. Typical equipment for the type of work that I do is you need environmental characteristics, of course, water, rainfall, wind, light, temperature. This machine up in the top left is called pressure bomb. You can use that to force water through the xylem in a plant and get an estimation of what the actual water potential is in the plant, so you get an idea of what water environment it's growing in. There's different times you can take this. You can take it during the midday, but as I'll show in a moment, that's sometimes unreliable. A really good way to do it is to lug that 50-pound machine out into a field at 4 o'clock in the morning. But as you can imagine, that's not very practical, but it is a good measure. To measure photosynthetic parameters, I use the top machine, which is a Lycor 6800. It's a portable photosynthesis machine. The gist of how it works is it takes air from the outside. It removes all the CO2, it removes all the water vapor, and then you can put back what you want. You can put water vapor in, you can put CO2 in, and then you pass that over the leaf surface. It has its own little light right here. And there's a chlorophyll fluorescence system in there as well. So you can get estimation of all your gas exchange parameters, all your fluorescence parameters. And then to fill in the gaps that we don't know, there's a few other methods like doing actual assays, I guess I'll call them, in a multimode plate reader like shown below. Get my cup of water real quick. These measures are always quite time consuming, and it requires a lot of technical expertise to be able to do these things. So they're not really useful as a management tool. For example, to be able to get information on the carbon reactions, that's after the primary photochemistry, you run through a process called an ACI response. CI is the concentration of CO2 inside the leaf, and as you can imagine, that's directly responsible for how much carbon is fixed, because you have to have a substrate for visco to act upon. This curve takes about 45 minutes to conduct if you're good, and then half the time you'll forget one little step and have to start over. But the blue line, the asymptote that approaches is the maximum rate at which Rubisco can fix carbon, okay? And the red line is effectively how much energy can the primary photochemistry actively produce to be able to run the carbon reactions. So that's VC max and J max. So when I talked about the primary photochemistry not showing a response, and it caused a big debate, the original theory that the light reactions are limiting our plants under water stress comes from this kind of data. So I went about trying to find a way to make it faster. Lycor did that for me. They created a new system. It's in Stenziano et al. 2017, where you can rapidly expose a plant to a range of CO2 concentrations. The dotted line up top, that's a traditional curve. The one below it is a curve that I did with a method called RACER. It's a rapid ACI response. And it knocks it down to about 7 minutes. And as you can imagine, that makes everything quite a bit easier. So to be able to use this technique in the field and try to get an estimation of what the underlying physiological mechanisms are and whether or not we can find other metrics that can be used as proxies for these measures so that we can actually get an idea of crop stress level. I planted 2 Gossypium species. That's Pima and upland cotton. And basically we set them into a well-watered condition and then followed them through a progressive drought, as it was like a 2 to 3 week drought. And we took daily RACER curves and survey measurements to be able to get all of our data, and then did some more lab data along with it, which I won't present today. So this is the result of 2 weeks of being in the field. It's 64 curves. It would have taken 48 to 52 hours to be able to get this data with one machine. With the more advanced techniques, you can knock it down to about 7. But then you're looking at 2 to 3 weeks of data processing, which we're working on streamlining as well. So back to this drought issue. Again, what metric should we use to indicate a moisture stress? Typically would be leaf water potential. I told you leaf water potential works really well before the sunlight hits the plant, because that's whenever you get the energy drive to start pulling water from the soil. It's through evaporation and being pulled up through the plant. Well, the left graph is net photosynthesis by midday leaf water potential. Midday leaf water potential is real finicky, and if you are in the southeast and a cloud goes over, you can change by 20% in a minute, just because that solar energy is not providing that evaporative power anymore. But there's another metric that I like to use more often is stomatoconductance. So on the bottom of a leaf or on the top as well, there's little holes, stoma. Well, depending on how open those are and the temperature inside the plant, you can change your transpiration. But there's a correction for that where you can just pick up what the aperture of the stoma is. And in my experience, anytime you have water stress related stress or events, stomatoconductance is always the one that really tracks really well with net photosynthesis, and that's the driver of what makes our cotton. So to kind of put that debate to bed about energy capture, I did a lot of work and basically regressing these parameters, which top left is net photosynthesis, and then you have CI. Remember, that's CO2 concentration inside the leaf on panel B. Panel C is J max, that's the maximum amount of energy going into the system for carbon fixation. Then you have VC max, that's think of it like the turnover rate for Rubisco itself. So it's how fast Rubisco can bind CO2 to RUBP. ETR is electron transport rate down at the bottom. If you remember back to the little thylakoid membrane that I showed you earlier, that's the rate at which that electron pushes itself through the entire system. So it's kind of an overall factor. And then the bottom one is actual quantum yield. So that's it's a proportion of light that makes it into the system, just a ratio. So if you look, we have a nice relationship with carbon fixation, CI, and things like that, but we don't have any relationship with the parameters calculated, like VC max, the turnover rate of the Rubisco is not being affected by drought. The amount of energy going into the system is not being affected by drought. So then the question comes up is, where does this energy go? Because it's apparently not making it into carbon fixation. Well, you can bust up electron transport rate with a lot of math. You have to have partial pressure information on CO2 and O2 and temperature. So those gases are differentially soluble at different temperatures. So it kind of it's a lot of math to get to this point, but you can bust it into what goes into actual carbon fixation and what goes into photorespiration. If you remember, photorespiration is a wasteful carbon event, or fixation event. And in upland cotton, you see a pretty good relationship with ETRP. And we know that from previous work that upland cotton tends to put a lot of excess energy into photorespiration to burn off all these processes. Peanut doesn't seem to do that. And, we're still looking at what's going on there. But what's really cool about all this and what makes it practical for us to use as a tool to gain information about the stress level of our crop and to be able to get to the point where we can use this for other purposes is panel 11. I still have it marked as 11. This would be panel B. One of those basic fluorescence signals is it's called basal fluorescence. It's F sub S whenever you pull it off your machine. As you get into a stress event, that signal goes down because that energy is being transferred into a process called non-photochemical quenching. It's basically the heat dissipation that I talked about earlier, like where the potential photon could end up going. It could end up going into photochemistry, reflectance, fluorescence, or heat. Okay. Well, as stomatoconductance decreases, so as you're going into a stress event, a water stress event, basal fluorescence rises. And it shows a lot of potential to be able to be used as a drone. Like let's go out and take your equipment and get a stress level. But let's say you have 100 plots or a 50-acre field or whatever. It's not practical. But it's also difficult to be able to get that data because you need a specialized piece of equipment. You have to bombard that system with a high light intensity and then be able to measure your signal. Well, currently we're working on a few things to make this a little bit easier. Right now, sun-induced fluorescence is a big deal. The person who really cracks this and makes it automated is going to never have to work again, probably. They basically fly a drone with a hyperspectral camera that can pick up the fluorescence signal at the particular wavelength we want. And you can use that to calculate that basal fluorescence. And so you can tell if this plant is coming under a stress event. It's still kind of tricky. It's a lot of math, and it takes people that are smarter than I am to pull it off, but maybe our new postdoc can figure this one out for me. A better method, in my opinion, is using spectroradiometer. There are labs that I'm working with, one in Tifton and one in Texas, and they are using this to detect nitrogen stress. And they get these plots with this huge variability, and they have ground-based spectroradiometers and then aerial spectroradiometers. And then they come in to try to see if they can pick up this stress. What's really cool is we think now that we can actually measure photosynthesis with an instantaneous point. You can go out with a spectroradiometer, click it, and you'll spend more time getting in and out of your side by side than you will actually collecting data. And once it gets to the point where we can do this drone-based, you can cover 5,000 acres in a day or whatever, and just have all that data. There's considerable processing, post-processing, as with any aerial-based stuff, but there still is a need to be able to use systems like I do to be able to correlate or to be able to calibrate these aerial-based systems and actually be able to pull this off. Because you have to make sense of everything. You have to know what signal's coming off to be able to know what to look at. But it's a pretty good time because our ability to collect data is just, the rate is just increasing exponentially. And I think I went through that faster than I intended, but do y'all have any questions? Right, it's hot.
The last one.
Mhm.
So if I understand correctly, the upland and Pima cotton,
Mhm.
the sort of metric for identifying drought, the stomatal conductance for both or for one of those?
Both of them relate. They both relate really well. It's just the fluorescence is different between the two because Pima has a different energy or a different stress response strategy. Cotton is, upland cotton is semi-homeothermic, so they seem to maintain a certain temperature, something below 32. But Pima tends to get a lot hotter, so that's why I included Pima because I wanted to know how it affects these parameters. But I didn't see any temperature increase in Pima. James Mahan did some research where he saw some excessive temperatures in Pima, is why we started going down that road.
So you think, like, based on, you mentioned the like Corpusco and not being competitively excluded by CO2 for like C3 versus C4, do you think stomatal conductance would be a good indicator of stress for C3 and C4? And then same kind of with that reflectance band with hyperspectral imaging. Do you think there may be different bands associated with C3 and C4 or even at the
I've steered
subspecies
clear
level?
from C4s because these techniques, like using an ACI response, doesn't really work for a C4 because they have that PEP carboxylase portion where they pull, they basically concentrate carbon and then separate out the carbon reaction so that they're in a non-oxygen, effectively a non-oxygen environment. So these kind of things don't really work for that. But the same process could work. If you go out and you have your hyperspectral data and then you have a stress indicator, stomatal conductance will be a stress indicator in a C4 as well. But I don't know.
Yeah.
Yeah.
But there is like a band associated with
The fluorescence band,
fluorescence.
yeah.
Okay.
I think it's 680 nanometers, but you can pick that up with any hyperspectral camera or any spectroradiometer. It pops out really well. That's effectively what a chlorophyll fluorometer is. It's like a really good photodiode for that one wavelength and a really bright light.
Okay.
Yeah.
That's cool. We have a spectroradiometer if we wanted to use one.
We have a spectroradiometer?
I have one. You can use it.
Can I borrow your
And I have a hyper,
spectroradiometer?
yeah, and a
I was gonna ask you
hyperspectral.
for one.
So,
Yeah. But,
Yeah.
we plan on, I've got some big studies going on in cotton to where we're looking at irrigation termination. And I don't know if you've seen cotton late in the season. If you don't water it long enough, it looks really terrible and producers are concerned. But, we're looking at whether or not the actual stress level is relevant. Like, is it hurting the crop? I don't think it is, but, with big studies like the 33-acre study I have, this sort of fly through with ground truthing calibrations would be a good opportunity to actually prove this concept and be able to use it for, rapid data
I
collection.
think so too.
Any other
Amanda,
questions?
can I ask a question from online?
Chat. Sassy is raising his hand again.
When you think about water conservation, you immediately go towards drought, but there's the other aspect of water logging, excessive water. As the climate changes, we'll have varying expectations and results when it comes to how water is distributed through the course of the year. Is there an application for the other side of the water world? Exploring the consequences from a photosynthetic position when it comes to excessive water presence?
I can't speak to cotton, but I can, it's a good question, by the way. I can't speak to cotton because I've been trying to get flooding research funded in cotton for my career, but nobody is interested in that. But I did get some research done in soybean, and soybean has a different stress response strategy. Like I told you, cotton burns off the excess energy, just kind of tosses it away because it's out there in high light. It's not limited by light, but species like soybean that have a little bit different evolutionary history, or they were probably like a more riparian plant that went through high and low light environments periodically. They don't have the same luxury, so they got to use what they can use, and so they'll, instead of burning off that energy, they'll just disassociate the antenna complex that sits on top of photosystem 2. It'll just disassociate and float off into the cell, into the stroma of the chloroplast. So you have different signals that you can pull that up with. So interestingly, though, your F sub S symbol will end up tanking as well, end up dropping off. So you can pick that up as well. For that one, I would suggest more of a hyperspectral approach, because you can also measure things like chlorophyll content through like various reflective indices like NDVI or red to green pixel ratios. But yeah, no, exactly. It's not just drought. It's any stressor. I mean, the one that's going on in Georgia right now, I'm involved more on the technical aspect of it. They're looking at nitrogen stress in corn. Like, what does it look like in corn whenever you have nitrogen stress? It yellows out. But you should be able to fly it to be able to get an estimation of midday photosynthesis with this sort of situation. I think it'll work for any stress, including your new hobby.
Sure.
Pathology.
Disease stress.
Yeah. But
Thank you.
yeah. And I have seen it work out for nematode stress in your advisor's lab too.
Oh yeah, we're gonna play with diseases there, you and I.
Yeah,
That'd be great.
How tolerant drought is cotton?
Cotton is considered a drought tolerant crop. It'll make cotton under a wider moisture range than a lot of crops will. That's basically what it boils down to. It's not inherently like better at it. It'll survive it. It's a perennial. It has other coping strategies that can help it, and has more control over that sort of thing. So yeah, in my opinion, yeah, it's drought tolerant, but in the degree that it will grow cotton with lower moisture, because it'll shed fruits and conserve energy to be able to do what it needs to do.
But with the view of like a drought, like a more temperature, there may be some kind of vulnerable to like some kind of insects with that.
I'm not sure your question.
There are some kind of insects, that worms in the cotton plants.
Oh yeah.
Is it a chance of higher chance of?
Yeah, does it make you more vulnerable to other pests and such?
Yeah.
I would probably say yes. I know in the terms of like, it can make it more difficult to manage pests like weeds. Because I know that generally speaking, if you put a plant under stress, it doesn't take up herbicides as well, and it doesn't translocate those herbicides to their actual active site as well. So yeah, I would assume, depending on the pest, like if you're looking at a soil pathogen like reniform nematode, which I have a lot of experience with, drought might make it better because, you know, they need a semi-aqueous environment to be able to survive. But from a weed perspective, it would counteract and make it more difficult to manage your pests. But yeah, that answer? John?
So you had mentioned that you could go about analyzing different parameters and different stressors. Is there a particular one that you're interested in, with the exception of drought? And the ones that you mentioned previously, heat?
Yeah, but heat and drought are so convoluted together, especially with the crops I've worked in. If you raise the air temperature in upland cotton, it's just going to transpire more water to cool itself. Like they have a limited homeothermy, and it's almost impossible to get a cotton plant over 35 degrees Celsius, to get the leaf above 35 degrees Celsius, unless you are also under a drought event, or depending on what stage of leaf growth you're at. I've taken cotton plants and cut them and then recut them underwater again and put them in a system where we have a recirculating bath to heat up these gas analyzers to where we can expose them to 45-degree air, and they'll stay at 32 because there's an unlimited water supply for them. But whenever you have a soil moisture event in an intact plant, you get a hormone response from the roots that ends up shutting down those stoma and decreasing your stomatoconductance. That's why it's a good reference parameter for drought in cotton. Other things will close stoma. Light quality can do it, but that's not a real big deal in the field. And in other plants, you can have direct drying of the stoma that can actually end up causing it, but cotton has a waxy cuticle that goes down the inside and prevents water loss from the stoma. So, but not, it's got to be drought and heat. Did I go over on time?
No, you're good. We're excellent. Yeah. Want to give Daryl a hand?